Hello, my name is Yaamr Ghanim. I'm in biomedical engineering department as a senior. As well, I do have a second degree from different country in chemical engineer. So before I'm gonna start explaining the posture, I would like to tell the story behind this idea because this is a novel idea and we are planning to apply for the PI of this idea because no one ever done this idea or tried to do it. So now in this poster we hide many things from that poster as an architecture design of the idea and many things because that's maybe gonna be a risk for the public disclosure. Like you're gonna risk the idea because that. So now the main part of this idea. Normally when we when we want to use a CRISPR technology, we need two parts. The first part is the Cas protein, our Cas, because we have many Cas protein like Cas9, 13, or other. And the second part is the guide RNA. So normally when we will transfect these two parts to the cells, these two parts gonna be active and will do the job assigned to the system. But the new design, the new idea it is re-engineered the guide. So now that guide, when it's going to be transfected with the Cas protein, the system will be inactive. That's the idea. So that system we transfect is the Cas with the new guide. It will be locked. It's not going to be able to work without our permission. So now our permission or the key for that lock is the microRNA. So when we will transfect a certain microRNA we design on this guide, then that guide can be active and do the job we want. As well, in the same way, that guide is now behave as a sensor for this microRNA. So when I proposed this idea in around May, I proposed other ideas, but this one was around May this year. Dr. Blair told me the idea is hard to test, so let me help you to find an easy way to test your idea. Then he directed me to use the DCAS system to test the idea. DCAS system is dead Cas protein. That Cas, it has the ability only to bind to the DNA, or DCAS13 can bind to the mRNA. So that Cas, it will not cut. Only it will have the ability to bind. Then the scientists add the VPR. Those the transcription part. Those gonna help to activate or suppress the transcription. Then we utilize this tools of the CRISPR technology with the idea. And in here in figure 3, now I'm showing why we are looking to test the level of microRNA, why we are looking for that sensor. Normally, that's because now many of studies showing when there is abnormal expression in the microRNA level, that will cause multiple cancer types, as well in the same time that can treat some of disease. So by that we were looking to find a sensor to test the microRNA level, and this is going to give us advantage to detect the cancer in the earlier stage, and thus can thus will help to find the treatment for that cancer before it's going to be developed and to the higher level. So here in the hypothesis part, in this pathway I'm showing, that's the pathway of the sensor. So now we need first we need the microRNA, then the microRNA will active the new guide, and that guide, when it's going to be active, will be react as the normal guide, So that's normal guide after is be active by the microRNA will go to the decas VPR plasmid, then bind to it. Then after these two bind, this will be bind to the CFV plasmid to activate the transcription with this plasmid. So after this binding, then we can see a CFV. So now there is a directional relation between microRNA and CFV. So now when we're going to have high microRNA levels, so we have to see high CFV level. By this way we can sense the microRNA for the new guide. So now the material and the methods. Normally I select to use the easiest way. The easiest way is by using the product from the PCR to test my new guide, and that's why I was able to produce a data in 3 months, because normally when people select the cloning path to do the testing of their ideas, only now to design one plasmid maybe can take like a month, less than that. So but now my new idea is test by using a U6 guide RNA. So that was like only running a PCR. Then after the PCR, this about the I transfected to the one of the type of the cells. I choose the SCT 116 with Jetoprime, and then this is the PCR product after is run on the gel. This of some, because as well here I want to mention I designed more than one guide, more than now almost more than 20, because I was wanna find which one is gonna be react more in positive way in our side, and what the difference between them, because I did some mutation on the re-engineering the new guide. This the equipment I used to collect my result. It was the first one was the fluorescent microscopy, then, and the second one was the flow cytometry. The result part here, I attached some image from the microscope to show the difference between here we have the positive control in the bright field, and here the CFV of the positive control. That's only a positive control, and the B1 showing the system, the complete all those 4 parts, microRNA and the other part, the complete system. This means with microRNA, this the bright field and the CFV level. We can see here there is a CFV level, and the C is the system without microRNA. Thus we can call it like a negative well, that it doesn't have the microRNA. So now we can see that there is less CFV here, and that's what thus is a proven the system is working. So here in the figure 7, in the part A, you can see the yellow column is the well without microRNA. It's everything, just all these 3 parts without the first one, the microRNA. And the blue one, the well with microRNA. So I test different design of the guide, but all of them showing in our hypothesis direction. There is like sensitivity to microRNA. So we were looking to see more CFP when microRNA there, because the microRNA will active the guide, and this guide can go and express a CFP. So now all of them showing in our direction more CFP with the one has microRNA, but as well we have like background, but now we are working to optimize this system and lower this background and showing the higher difference. But here now while I was working, I tried to play with the ratio of the microRNA to the guide. So you can see like some of this ratio is showing more different between the one has microRNA to the one without microRNA. So that's why now we are trying to optimize by using different ratio, as well to re-engineer some of or do some mutation on the guide. And here I titrate guide number 1, and you can see now there's the x-axis is the ratio between microRNA to the new guide. So now 1 microRNA to the 2 ratio of guide, 2 ratio of microRNA to 1 ratio of guide, and you can see this the column is the different in our side. This means the difference is in the CFP with the well have microRNA to the well without microRNA. So now when the difference going to be higher, this means the system is improve itself. It this is a positive point for us. So now we working to optimize that. And the figure 9, that's the flow data. That's data from the flow. All this data from flow, but this is the normally figures we are using to draw our data after we are in the flow. So now the red one is the positive control, and the blue one here, the one the system, all the system with FF3, everything is looking now, we can see is higher than the yellow one without FF3, and the green one is the negative control. So now all of them they show like the blue is higher than the yellow. This means is that our system sensing the microRNA. And then now conclusion of this idea. Before this idea, the Cas system was free to do its job inside the cell. So now when we transfect the Cas protein with the guide to the cells, it has the freedom to do whatever we assign that system to do. But now inside the cell with the new guide, when we're gonna transfect them inside the cell, now this system is not gonna be active without our permission to let that system active. This means after we're gonna transfect the microRNA, then this microRNA will free the system and make the system work. So now the only, I think this is the only time we were able to control the Cas system inside the cell, and in the same way we are sensing another structure by the system. So that's why this is a novel idea, and no one ever done it before. So now I hope this idea gonna go further, and we can do a lot of thing with it, like a treatment, sensing, and anything of this part. So now, during that time, I'm working to optimize the signal, like by sending the background signal to the zero, this the yellow signal to the zero, and make the difference higher. And as well, we are planning in the future to build in one system a plasmid, has everything, has the guide, the micro, all sort of has the guide, and has this guide gonna sense multiple microRNA, as well as the for example the Cas9 VPR and the fluorescent for this microRNA. So yeah, we are looking to do a lot of things with this new technology. I can say it, because this is I think is a new technology to deal with the CRISPR. So yeah, that's everything, and I would like to thank the member of the Players Lab, because they helped me a lot to learn the basic of the biology and how to run the devices there, and how to do some of the kind of cloning and that stuff. Yeah, first one, Dr. Lee, and then Tech and Chance and Stone and John, all of them helped me with that, as well Ahmed, he helped me with that. So I would like to thank them, and especially I have to thank Dr. Blair's, because he gave me this opportunity to test my idea, and and at the same time I want to thank him because he believed in me and my idea gonna work. Because the first time he was keep asking me, I wanna resource, I wanna find how your idea gonna work. I told him, this is a new idea, I cannot find any resource to do that, so you just need to believe me and let me do it. Then he did that, and I was lucky. After the, I think I want to say it, after the God support, because that's this idea is, I don't know how it's come to me, but I think there is some of the mercy from the guy, the God, and he provided me this idea. Thank you.